Assalamu alaikum. I am Dr. Sidra Arshad, Assistant Professor of Physiology, and today I shall be walking you through the physiology of thyroid metabolic hormones. This lecture is the third in series of our crash course. Okay, so let's just start. For each and every major hormone, there is this scheme that you can follow to organize the information. For each and every hormone, you have to start by looking at its chemical composition. What is the chemical nature of that hormone? This will help you to understand some of its characteristics pertaining to its synthesis and release, then how that particular hormone is transported in blood. Is it free? Is it bound to proteins? Or is it transported in both ways? Then when you know the chemical nature of this hormone, you shall be able to tell its mechanism of action at the cellular level. For example, there are certain hormones which involve the genetic machinery of the cell. Their action involves the activation of the genetic machinery of the cell. And then there are certain hormones whose receptors are present right on the cell membrane, and then they involve the second messenger systems. Then afterwards, you are going to follow the system-wise scheme to see the effects of that particular hormone on each and every target tissue. The next step is to know the regulation of that hormone. What are the factors which are stimulating the release of that hormone? What are the factors which are inhibiting the release of that particular hormone? And how the basal level of that hormone is regulated? What are the feedback mechanisms involved? Afterwards, you are going to apply this information to the clinical settings. What exactly can be the abnormalities of that particular hormone? Endocrine physiology, there can be two abnormalities. Either they are going to be excessive levels of that particular hormone, or the hormone is going to be deficient. So this scheme can be used for each and every hormone, and today we are going to use it for the thyroid hormone. We're going to begin with the synthesis and release. Then we are going to see the transport characteristics of the thyroid hormones. Then we are going to find out what is their mechanism of action, how do they operate in the cell. Then we are going to see their effects on multiple tissues in the body. We are going to see them one by one. And then we are going to find out how the regulation of thyroid hormone occurs in the body. And then we are going to find out the abnormalities. How do they affect the human body? So let's start with synthesis and release. But before that, there is going to be a little overview. So thyroid gland, it is located below the larynx in front of the neck. It produces two major hormones, T4 and T3. T4 is also known as thyroxine and tetraiodothyronine, whereas T3 is known as triiodothyronine. Thyroid gland secretes these two hormones. However, its major output, about 93% of the hormone that is released from the thyroid gland is actually T4, and only 7% is released in the T3 form. However, in most of the peripheral tissues, the T4 is converted into T3 because T3 is about 4 times as potent as T4. So most of the actions of thyroid hormones are actually mediated by T3, and T4 undergoes peripheral conversion into T3. What exactly is the function of thyroid hormones? In a nutshell, they profoundly increase the metabolic rate of the body. They have a marked effect on the metabolic processes at the cellular level. They tend to increase the metabolic rate of the body. So this is the information about T4 and T3. We are going to see their characteristics one by one, and then we are going to have some features to set them apart. This is the structure of the thyroid gland. Thyroid gland is composed of follicles. Multiple follicles are found in the thyroid gland, and in between these follicles, there is a rich blood supply. These follicular cells, they are responsible for the synthesis of thyroid hormones. You can see there is right in the center of the follicle, there is a substance known as colloid. This colloid is rich in proteins, and it actually takes part in the synthesis of thyroid hormones. Lining this colloid are the follicular cells. These follicular cells are the site of synthesis of thyroid hormones and also the site of release. The hormone which is synthesized in these cells is released into the closely associated capillaries, and this is how these hormones travel in the blood once they are synthesized in the follicular cells. Also, in association with follicular cells, very close to it, you are going to see another population of cells which are known as C cells. These C cells, they produce another hormone which is known as calcitonin, which is involved in the calcium homeostasis. So today we are going to focus on the thyroid hormones themselves. So these follicular cells, these are cuboidal epithelial cells, and they are responsible for the synthesis of thyroid hormones. Now this is very important. One of the most important questions, frequently asked questions, is the steps of thyroid hormone synthesis. So you have to remember these steps and you have to remember them in a proper sequence. So most of you know the association of thyroid hormones with iodine. Iodine is required for the synthesis of thyroid hormones. This is one of the main compounds which is required for the synthesis of thyroid hormones. So right here in this diagram, you can see the structure of a follicular cell. So this is a follicular cell. Follicular cell has two sides. The side which faces the colloid is known as the apical side, and the side which faces the blood vessels, it is known as the basal side. Determining the polarity of the cell over here is very important because there are certain transport mechanisms which are taking place in the apical side, and then there are certain transport mechanisms which are taking place at the basal side. After you have determined the polarity of the follicular cell, now we are going to begin with the steps. You have to see the number which follows the sequence. So first step is iodide trapping. We have the iodide in the blood obtained from the food sources. Now we have to trap this iodide into the cell for the synthesis of thyroid hormone. So this is the very first step. How it is going to take place, if you see right over here on the basal side which is present as association with the blood vessels. On this basal side, there is a transporter which is going to help us in trapping the iodide. This transporter right over here, you can see the sodium potassium pump, and very next to the sodium potassium pump, there is the iodide trapping mechanism. So the number one step is the iodide trapping. Iodide is trapped by a special transporter, which is known as the sodium iodide symporter, NIS. And for sodium, I for iodide, and S for symporter. This is the sodium iodide symporter. What does it do? For every 2 sodium ions that it takes up, it transports 1 iodide ion into the follicular cell. The gradient for this transport is established by the sodium-potassium ATPase. So the number one step is iodide trapping. Iodide, which was present in the blood, obtained from many food sources, has now been trapped into the follicular cells. Where is the iodide right inside of the cell? The second step, we have to transport this iodide into the colloid for the synthesis of thyroid hormone, which is going to take place in the colloid. So how it is established? The transport of iodide ion out of the follicular cell is established by a protein. A protein takes part, a protein transporter is involved right over here on the apical side, which is known as pendrin. Pendrin protein, it is actually a counter transporter. It counter transports chloride and iodide. So this counter transporter is located on the apical side. So now we have 2 types of transporters, one on the basal side, which actually helped us trap the iodide, and another transporter which was located on the apical side, which helped us move this iodide into the colloid. So after the iodide has moved into the colloid or out of the follicular cell, it has to be converted into the iodine. Iodide cannot directly combine with tyrosine to make the thyroid hormones. It has to be oxidized. So for this oxidation, closely associated with the apical membrane are peroxidase enzymes. These peroxidases, they oxidize the iodide ions and generate the hydrogen peroxide, which is promptly catabolized. So this is the third step, oxidation of iodide ions. After the oxidation of iodide ions has taken place, there is going to be a step which is known as organification of thyroglobulin. Now, the synthesis of thyroid hormone is quite interesting because it is taking place on a protein which is known as thyroglobulin. Thyroglobulin is a protein which has multiple tyrosine units. The iodine that has now been oxidized from the iodide ions, it combines with the tyrosine residues of the thyroglobulin molecule. And this process is known as organification of thyroglobulin. So once the organification of thyroglobulin has occurred, is going to take place. That is the synthesis of thyroid hormone. So the fourth step is known as the organification of thyroglobulin, which involves the combination of iodine with the tyrosine residues of thyroglobulin. We are going to see the detail how the thyroid hormones T3 and T4 are actually synthesized just in a bit. After these hormones have been synthesized, you can see the TG over here, it represents thyroglobulin. These hormones are present in association with thyroglobulin, T3, reverse T3, and T4. These three hormones. What is reverse T3? We are going to find out in just a bit. So now this entire thyroglobulin, thyroid hormone complex has to be taken up into the cell once again to be released into the circulation. This is accomplished by two processes. Number one process is simply the pinocytosis of the colloid droplet, which contains the thyroglobulin molecule and associated thyroid hormones. Once it is endocytosed by pinocytosis, this colloid droplet, you can see right over here, it has reached inside of the cell. Now it is present in the cytoplasm of the follicular cell. Once the colloid droplet is inside of the cell, there is another process, which is the transcytosis, once again, where the thyroglobulin molecule, it binds with the megalin protein, and this megalin protein and thyroglobulin complex is internalized by the endocytosis, and it moves out of the cell in the similar fashion. So there are two mechanisms. One is simply the pinocytosis of the colloid droplet, which contains the thyroglobulin molecule associated with these thyroid hormones. And the other one is the binding of thyroglobulin with this megalin protein present on the apical cell membrane of the follicular cell. And this complex is then internalized. So these two steps are how the thyroid hormones are internalized into the cell. So once the colloid droplet is present inside of the cell, what is going to happen? The lysosomes are going to fuse with this colloid droplet. Why it is important? If you remember that the thyroid hormones, they are not free. They are right now associated with the thyroglobulin molecule. They have to be broken free from this. So how it is going to be established? It is going to be established by proteases, which are going to be present inside of the lysosomes. So these lysosomes are going to fuse with the colloid droplet. And once this lysosome and colloid droplet combination has formed, it is going to, proteases present in the lysosome are going to digest these or separate this, cleave the hormones from the thyroglobulin molecules. These proteases, which you can see right over here, these are present inside of the lysosomes. Here, a step you can add, that is the fusion of the lysosomes with the colloid droplet. This is going to cleave the thyroxine and triiodothyronine, that is T4 and T3, from this thyroglobulin molecule, which can then be released into the blood. So the thyroid gland releases T4, T3, and reverse T3 into the circulation. This is the sixth step. Seventh step is the removal of iodine from monoiodotyrosine and diiodotyrosine. How is this established? You can see associated with the thyroglobulin molecules were other compound MIT and DIT. We are going to see what these compounds are. So these are recycled. We have to conserve the iodine. So once the T3, T4, and reverse T3 have been released, these MIT and DIT, they are not released. They are actually deiodinated, and the iodide ions are cleaved for them, and they can be used for the synthesis of thyroid hormone in the next cycle. So this is how the synthesis of thyroid hormone is taking place inside of the follicular cells. So the seventh step was deiodination, which helps us recycle the iodide ions. So right now you can pause this video and you can test yourself for the step of synthesis and the mechanisms involved. So let's just see them in the further detail. Number one step was iodide trapping into the follicular cell, which was established by sodium iodide co-transporter on the basolateral side. Second step was transport of iodide ion out of the follicular cell. It was transported out of the cell by the pendrin transporter, which was actually the chloride iodide antiporter. Third step was oxidation of the iodide ion. Oxidation of the iodide ion was aided by the hydrogen peroxide with the help of peroxidases enzymes, which were associated with the apical membrane of the follicular cells. Why was this important? Because iodide ion cannot combine with tyrosine on the thyroglobulin molecule, but the iodine can combine. So it had to be converted into iodine. It had to be oxidized. So oxidation was taking place with the help of peroxidase enzymes, which were associated with the apical membrane. The next step was organification of thyroglobulin. What was organification? It was binding of iodine with the tyrosine of the thyroglobulin. This is the organification. These reactions were taking place. If the iodine, it combines with tyrosine, it forms monoiodotyrosine. Monoiodotyrosine could then combine with diiodotyrosine and form triiodotyrosine, or diiodotyrosine plus diiodotyrosine. If they combine, they are going to form thyroxine or tetraiodotyrosine. If diiodotyrosine combines with monoiodotyrosine, it forms reverse T3. So MIT plus DIT, it forms triiodotyrosine. But DIT, if it combines with MIT, it forms reverse T3. So the major output is T4 and T3. Reverse T3 जो है, you can ignore it for a while. The next step is penocytosis or colloid resorption. So now the hormones, they are formed, but they are present in association with thyroglobulin molecule, which has to be internalized. So a colloid droplet containing thyroglobulin in association with T3, T4, reverse T3, MIT, and DIT is internalized. This internalization was established by penocytosis and the combination of thyroglobulin molecule with the megalin protein. Once they are internalized, the T3 and T4 have to be broken free from the thyroglobulin molecule, and this is done by the proteases which are present inside of the lysosomes. They cleave the T3 and T4 from the thyroglobulin. Once the T3 and T4 have been cleaved, they are released into the circulation, while the MIT and DIT, they undergo deiodination, which is the removal of iodide ion from these two compounds. And this is how iodide ions are recycled to be used for the synthesis of thyroid hormones in the next cycle. So these are the steps of synthesis which you have to remember. If you follow this sequence, you are going to be sure of not missing out on any steps. Just to reiterate the importance of iodine pertaining to the synthesis of thyroid hormones. So the dietary iodine, which is taken up and reabsorbed, the dietary iodine which is absorbed from the GIT, it is going to be used up by the thyroid follicular cells. Most of this iodine is actually excreted by the kidneys. However, some of that iodine is taken up by the thyroid glands, and the thyroid glands, they also end up excreting some of the iodine, but they actually use this much of the iodine for the synthesis of thyroid hormones. So you can see that the most of the dietary iodine is in fact excreted out of the kidneys, but the iodine which is actually being used is being used by the thyroid gland for the synthesis of thyroid hormones. Okay, now we have seen the synthesis of thyroid hormones. Let's just see how these thyroid hormones are transported in the blood. So the output of thyroid gland is T4 and T3 majorly. So most of the T4 and T3 which are produced, they get bound to the proteins. These are small molecules. They have to be trapped in the circulation, which is established by them being bound to the proteins. The hormones which are going to bring about the physiological actions, they are going to be free hormones, because only the free hormones can bind with the receptors. However, there is a circulating pool of these hormones, and this circulating pool is established by the proteins which help them trap these hormones. So T4 and T3 are bound to the proteins. Which proteins are involved? Most of the hormones are bound to the thyroid binding globulin. Thyroid binding globulin is a protein which binds the thyroid hormones. So they are 99% bound and only 1% they are in free form. So 99% bound thyroid hormones are bound to thyroid binding globulin, a lesser amount to transthyretin, and then to albumin. So these are the proteins which are involved in the binding of thyroid hormones. Most of the thyroid hormones, they are bound to thyroid binding globulin. Some are bound to transthyretin and very lesser amount to albumin. So this is how thyroid hormones are circulated in the blood. Only 1% free and 99% bound to these proteins. Once the thyroid hormones, they reach their target tissue, what they are going to do. So we are going to follow this diagram to see the steps that how the thyroid hormones bring about their actions. So number one step is the movement of these hormones to their receptors. This is the cell membrane. You can see over here T4 and T3. They are taken up by the target cell. Once they are present inside of the cytoplasm, there are certain transporters involved over here. Once they are present inside of the cytoplasm, as you remember that T3 is the main active hormone, which is most potent one, which does actually bind to the receptors. So the T4, which was more in amount in the circulation as it was released by the thyroid gland in the greater amounts, it has to be converted into T3. It is established by these deiodinases enzymes. These deiodinases enzymes, these are known as thymidine specific deiodinases. So number one step, you have seen the thyroid hormones, they are taken up into the target cell and then they have to go to their receptor, which is present inside of the nucleus. So these thymidine specific deiodinases, these are present inside of the many cells of the body and they exist in three isoforms, D1, D2, and D3. D1 or deiodinase 1 is found in most of the cells, most of the tissues, including the thyroid tissue itself. D2 is found in the cells of the CNS, and while D3 is found in the reproductive organs. So these deiodinases, they convert T4 into T3. T3 then moves inside the nucleus, and its receptor is located on the DNA. The thyroid hormone receptor is present in association with another protein, which is known as retinoid X receptor. Once the T3, it binds to the thyroid hormone receptor on the thyroid hormone response element, which is a gene sequence. What does it do? It activates the gene transcription. Multiple proteins are then synthesized, and the synthesis of these proteins, they are going to affect the many systems of the body, which include growth, CNS development, cardiovascular, and metabolic systems. So what exactly happens is this: the thyroid hormone, when it binds with these receptors, it brings about transcription of many genes, which ends up synthesizing many proteins, which are going to bring about the effects of thyroid hormones. So this is the mechanism of action of thyroid hormones. You have to remember this step, that most of the T4 is to be converted into T3 as it binds more actively with its receptor, and it is T3 which is going to bring about these effects eventually. So T4 has to be converted into T3. It is more potent. So now there are certain characteristics of T3 and T4 which you can discern. If you have to compare them, you can see that the major output of thyroid gland is in the form of T4 and T3. T4, 93% is T4 and 7% is T3. Their binding characteristics are a little bit different too. T4, it binds more avidly with the proteins, while T3 binds less avidly. And then T3 is about 4 times more potent than T4. And it is the T3 which is bringing about the actions of the thyroid hormones. Now that we have discussed the mechanism of action of thyroid hormones, let's discuss the physiologic effects of thyroid hormones on the tissues. We are going to look at them one by one. Let's start with the cellular metabolic activity. So thyroid hormones, they increase the cellular metabolic activity. They are involved in increasing the basal metabolic rate of the body, which is brought about by increasing the cellular metabolic activity. So what do they do at the cellular level? They increase the number and size of mitochondria so that the ATP production is increased. And then they increase the utilization of that ATP itself so that ATP is cleaved, energy is released, and also the heat. So they increase the activity of sodium-potassium pump. They also increase the membrane leakiness to sodium ions, which in turn increases the activity of sodium-potassium pump by establishing the sodium gradient. As far as the metabolism is concerned, we are going to look at the 3 metabolites one by one. Let's start with the carbohydrate. So the thyroid hormones, they increase all aspects of carbohydrate metabolism. They increase all the facets of carbohydrate metabolism, be it glycolysis, glycogenesis, glycogenolysis, gluconeogenesis. So they increase the glucose levels and its utilization. How do they do that? They increase the cellular metabolic enzymes. So the more and more enzymes are made available for the carbohydrate metabolism. The enzymes which are involved in glycolysis, for example, their synthesis is increased. They also potentiate the effects of other hormones, the hormones which are involved in bringing about the metabolic effects. Their actions are potentiated in the presence of thyroid hormones. For example, the thyroid hormone, they increase the release of insulin. Insulin in turn increases the glucose uptake by certain cells. As far as proteins are concerned, the thyroid hormones, they increase the breakdown of proteins more so than their synthesis. Thyroid hormones, they potentiate both the breakdown and the synthesis of proteins. However, their main actions pertaining to proteins is catabolic. They potentiate the proteolytic effects of other hormones. This effect is also brought about by the thyroid hormone's effects on the other hormones' actions, which is known as the permissive effect. As far as lipids are concerned, they mobilize and degrade the lipids. This is the main lipolytic effect. They increase the synthesis of fatty acids in the liver. How do they do that? They decrease in the body pool and plasma levels of total cholesterol and LDL cholesterol. Levels of these types of cholesterol are decreased. They also increase the formation of LDL receptors. So, metabolic rate is increased by the thyroid hormones. They increase the metabolic rate by 60 to 100% above normal in certain cases, and they also stimulate thermogenesis. They increase the ATP utilization, especially in skeletal muscles, where there is increased activity of sodium-potassium pump and sarcoendoplasmic reticulum-associated calcium ATPase. So by increasing this activity, they are actually increasing the utilization of ATP, and this utilization of ATP is going to produce heat as well, and energy is released. So this is the mechanism of increased metabolic rate. An example is given for the skeletal muscles. They also stimulate thermogenesis, especially in the brown fat, which is present in the neonates and infants, not in so much in adults. In some lean individuals, it can be found in the mediastinum. And here they stimulate the synthesis of uncoupling protein 1, which is involved in decoupling the oxidative phosphorylation so that ATP is not produced, but a lot of amount of heat is generated. So this is how this hormone is involved in the thermogenesis by the direct production of ATP and its cleavage to release heat, for example, in skeletal muscles, and then its actions in the brown fat. Now let's look at the cardiovascular effects of the thyroid hormones. Clinically, the effects of thyroid hormone on the cardiovascular system are very important. The thyroid hormones, they exert the following effects. They increase the resting heart rate, stroke volume, speed of contraction, force of contraction, and they shorten the diastolic relaxation time. So all of these effects, they actually increase the cardiac output of the heart. And these effects are mediated by direct and indirect actions of the thyroid hormones. What do they do? Directly, the thyroid hormones have a permissive action towards the actions of catecholamines. What does that mean? That catecholamines, they exert their effects only in the presence of thyroid hormones. The maximal effects are exerted only in the presence of thyroid hormones. Why is that? Because the thyroid hormones, they actually increase the number of beta adrenergic receptors. So when the number of beta adrenergic receptors is increased, the response to circulating catecholamines is enhanced. They also increase the proportion of alpha myosin heavy chain. This type of chain has a higher ATPase activity. They increase the expression of sodium-calcium exchanger and decrease the expression of SERCA, so that the force of contraction is increased. So the indirect effects, we have seen the direct effects. The indirect effects are brought about by the overall effect of thyroid hormones on the metabolism. As they increase the heat production and cellular metabolism, as a result of that, there is carbon dioxide production in the tissues. So carbon dioxide and the increased heat, it is going to cause vasodilation, and this vasodilation is going to decrease the systemic vascular resistance. Decrease in systemic vascular resistance results in decrease in diastolic blood pressure, and when this blood pressure is decreased, there is marked sympathetic discharge. So there is reflex increase in adrenergic stimulation. So both of these indirect effects and direct effects, they are going to increase the cardiac rate and output and increase blood volume. So the patients of thyrotoxicosis, they are prone to develop high output cardiac failure. As far as respiratory system is concerned, the thyroid hormones, they increase the respiratory rate and depth of breathing. They also increase the 2-3 ppg levels. All of these are involved in greater transport of oxygen to the tissues, which are utilizing oxygen more and more now because of the higher metabolic rates. So these respiratory effects are occurring in response to increased oxygen utilization at the cellular level. The effects of thyroid hormone on CNS development are quite important, especially during the fetal life. They regulate the timing and pace of development of the CNS in fetal life. And in normal day-to-day life, they enhance wakefulness, alertness, responsiveness to various stimuli, auditory sense, awareness of hunger, memory, and learning capacity. And they're also involved in mediating the normal emotional tone so that patients of thyrotoxicosis or hypothyroidism, they can present with psychological symptoms. What are the mechanisms which are involved during the fetal life? There is the development of CNS. Thyroid hormones, they promote proliferation of axons and branching of dendrites. They potentiate synaptogenesis, formation of synapses, that is, myelination and cell migration. They also increase the rapidity of celebration, that is the thought process formation. So this is the these are the actions of thyroid hormones on the CNS. As far as skeleton and muscles are concerned, in the fetus, thyroid hormones are important for bone formation, and at the time of puberty, they are involved along with the growth hormone in bone growth, and they are also important for the growth of soft tissues and functions of skeletal muscles themselves. So if the person is deficient in thyroid hormone, the bone growth can be affected. We are going to see it subsequently in the disorders. How do these actions occur? The thyroid hormones, they promote endochondral ossification and linear bone growth. They cause maturation of the epiphyseal bone centers, and together with the estrogen, they bring about the closure of the epiphysis around puberty. They promote endochondral ossification, linear bone growth, bone resorption, and maturation of the epiphyseal bone centers. In the gastrointestinal tract, they have these effects. They increase the rate of carbohydrate absorption. As we had already seen that they increase almost all the facets of carbohydrate metabolism. The number one step is to increase the actual uptake or rate of carbohydrate absorption from the gut. They increase GI secretion and motility. All of these effects, they are occurring in response to increased appetite and food intake, which is brought about by the increased cellular metabolism. Thyroid hormones are also important for the functioning of reproductive system. They are involved in maintaining the fertility and libido in men. In females, they are involved in the regulation of reproductive cycles, regulation of ovulatory cycles, so that in the patients who have hyperthyroidism, there can be amenorrhea or scanty menstruation. This is a bit of a paradoxical sounding effect that this is hyperthyroidism, but what happens is that the cycles are not regulated. Ovulatory cycles, they are disrupted. So amenorrhea or scanty menstruation may occur, while in females who lack in thyroid hormones, who have low levels of thyroid hormones, heavy menstrual bleeding may occur. So the thyroid hormones, they have a permissive role in regulation of reproductive function. How do they bring about these effects? They actually alter the metabolism of steroid hormones. That is, they stimulate hepatic synthesis and release of sex steroid binding globulin. This is one of their effects, that they increase the synthesis of sex steroid binding globulin, which sequesters these hormones, reproductive hormones. So this is one of the possible mechanisms. The thyroid hormones, they also exert their effects on other endocrine glands by promoting secretion of several hormones, for example, insulin or parathyroid hormone when they are involved in the bone growth and resorption. They also cause inactivation of steroid hormones because they increase the metabolism. So the inactivation of steroid hormones could actually occur by increasing their metabolism, which as a result is going to stimulate the release of ACTH from the pituitary, and the ACTH is going to stimulate the release of glucocorticoids. So even if they're causing the inactivation of hormones, there is going to be a feedback increase in those hormones. So that is why the thyroid hormones, even if they are increasing the metabolism of some steroid hormones, the levels of those hormones, they do not markedly decrease because of the thyroid hormones. So these were the actions of thyroid hormones on some major organs. Now we are going to see that how the secretion of thyroid hormone is regulated. So the regulation of thyroid secretion, it occurs at 3 levels. In order of importance, you may say that the hypothalamo-pituitary thyroid axis, it regulates via feedback mechanisms the release of thyroid hormones. Then the thyroid secretion, it is also influenced by the effects of body temperature and some other neurogenic stimuli. The release of thyroid level at the level of the gland itself can also be regulated by the iodine. So let's just look at them one by one, starting with the hypothalamo-pituitary thyroid axis. So what happens is that in response to certain stimuli, for example, decreased temperature, the thyrotropin releasing hormones, they are released from the hypothalamus. TRH, thyroid releasing hormone, is released from the hypothalamus. It acts on the anterior pituitary cells, which are involved in the production of TSH. These cells then release TSH or thyroid stimulating hormone. The thyroid stimulating hormone has its receptors on the follicular cells of the thyroid gland. It binds with those receptors, and increase in the cyclic AMP levels brings about certain effects in the thyroid gland. And these effects are, there is increased proteolysis of thyroglobulin, so that more T3 and T4 is being made available to be released. There is increased activity of iodide pump, so that more and more iodide is being trapped and used for the synthesis of thyroid hormones. There is increased iodination or organification of thyroid hormones. There is increased size and increased secretory activity of the thyroid cells. So you can see they are almost increasing all the aspects of thyroid hormone synthesis, as well as increasing the size and increased secretory activity of the thyroid cells. This increase in size, we are going to see the significance of that when we'll be discussing the disorders shortly. They also increase the number of thyroid cells. So almost all aspects of thyroid hormone synthesis are facilitated or increased by TSH. Now, once the T3 and T4 levels, they are increased, they have a negative feedback effect on the release of TSH. They inhibit the release of TSH, and T3 and T4 also inhibit the release of TRH. So this is the negative feedback effect of these hormones. So for the hypothalamo-pituitary thyroid axis, begin with the release of TRH from the hypothalamus in response to certain stimuli. TRH's effect on the TSH secreting cells of anterior pituitary, TSH's effect on the follicular cells, and then the negative feedback effect of T3 and T4 on the TSH and TRH. So there are certain stimuli which can affect the release of TRH. We are going to see what are those. So exposure to cold. Exposure to cold, it causes the hypothalamic centers for temperature control to become activated. And these centers, then they increase the release of TRH, which is going to stimulate the release of TSH, and TSH is going to increase the secretion of T3 and T4 hormones, which is going to bring about increase in metabolism, production of heat, and thermogenesis to counter the drop in body temperature, as well as prolonged fasting. What prolonged fasting does, it decreases the leptin level, and these decreased leptin levels can affect the hypothalamic centers, which then respond by decreasing the release of TRH. This is going to conserve the body stores of energy so that not much energy is being spent in the scenario of prolonged fasting or starvation. And another scenario is when there is increased stress levels, emotional states, or anxiety, there is increased sympathetic activity, which actually increases the metabolic rate and produces more and more body heat. So the hypothalamic centers for temperature control are once again, they detect this body temperature and then they decrease the production of TRH so that more heat is not produced. So this is the regulation of release of TRH by these three other stimuli. These are exposure to cold, prolonged fasting, and then there is this anxiety, stress, and emotional states. So basically, these are the stimuli for the release of TRH. Either the release of TRH is stimulated or inhibited pertaining to the different scenarios. So autoregulation of thyroid gland function is caused by iodide itself. So we have seen the hypothalamic pituitary thyroid axis. We have seen certain stimuli which can affect the hypothalamic release of TRH. Now we are going to see the regulation of thyroid hormone release at the level of thyroid gland itself. So it is regulated by the iodide ions. Iodide ions, they exert a biphasic action. What is that biphasic action? It means that at the low levels of iodide intake, the rate of thyroid hormone synthesis is directly related to the availability of iodide. That is, when the iodide levels are normal at the lower range, what happens is that the thyroid hormones, they are synthesized. When there is iodide present inside of the cells, it is almost directly proportional to that. However, at excessive iodide intake, the intraglandular concentration of iodide reaches such a level that there is a paradoxical suppression of thyroid peroxidase activity. When the thyroid peroxidase activity is paradoxically suppressed by these excessive iodide concentrations inside of the follicular cell, what will happen that the synthesis of thyroid hormones will be decreased. So this is the biphasic action, that at the lower quantities, the synthesis of thyroid hormones is proportional to the presence of iodide ions inside of the follicular cell. However, if the iodide ions reach an excessive concentration, there is a paradoxical suppression of thyroid peroxidase activity. So the synthesis of thyroid hormones is actually decreased. This can be applied to an effect which is known as Wolff-Chaikoff effect. What actually happens is that there is a temporary inhibition of thyroid hormone synthesis and release in response to high levels of circulating iodine. In certain iodine deficient individuals, if they are given exogenous iodine or if it is replaced somehow, what happens that all of a sudden there is now excessive quantity of iodine which is available. This iodide, when these levels increase in the cell, that it accumulates inside of the follicular cell, it inhibits the activity of thyroid peroxidase. So what happens, you have provided the iodine to an iodine deficient individual, but instead of increased biosynthesis of thyroid hormones, it is actually being decreased. So this is known as Wolff-Chaikoff effect. This decrease is temporary, however, because in response to excessive iodine levels, what happens is that eventually the sodium iodide symporter, it becomes down-regulated. The level or the number of those transporters on the basal side of the cell, it decreases. And what happens is that the iodide concentration inside of the follicular cell, it actually drops as a result. So the synthesis of thyroid hormones then increases because This inhibitory effect of excessive iodide ions on the thyroid peroxidase is then lost. So high concentration of iodides, about 100 times the normal concentrations, they can exert an anti-thyroid effect. They can act as an anti-thyroid substance. They reduce the iodide trapping, they inhibit the endocytosis of colloid, reduce the size of the gland, and decrease in blood supply of the gland. So these are some of the other effects of high concentration of iodides. What they do is that they reduce iodide trapping, inhibit endocytosis of colloid, reduce the size of the gland, and decrease in blood supply of the gland. There is a clinical importance of that in patients who are undergoing thyroid surgery. Before that surgery, the high concentrations of iodide are usually administered to those patients so that especially their blood supply to the gland is decreased to prevent the blood loss during surgery. So before discussing the abnormalities of thyroid glands, let's just discuss the effects of hypothyroidism and hyperthyroidism on the body. If you already know the physiological effects of these hormones on the systems, you can predict what is actually going to happen in both of these cases. So in hypothyroidism, the individual is cold intolerant because the basal metabolic rate, the body is now reduced. There is less heat production. The person is intolerant to cold because the heat generating mechanisms are not working. There is increase in weight gain because of this reduced metabolism. In hyperthyroidism, however, there is heat intolerance because of the excessively elevated metabolic rates of the cells. There is sweating and weight loss because of the burning of the calories. As far as skin and hair changes are concerned, in the hypothyroidism, dry, cool skin with coarse and brittle hair is seen. There is diffuse alopecia, brittle nails, puffy face, and generalized non-pitting edema. We are going to see the mechanism of that shortly. In hyperthyroidism, the skin is going to be warm, moist, fine hair, onycholysis, pretibial myxedema in Graves' disease. We're going to see what exactly is Graves' disease and why these features are actually occurring. But you can just know right here that in hypothyroidism, there is vasoconstriction at the level of skin arterioles, and in hyperthyroidism, there is vasodilation. So some of these features can be explained by these two phenomena. As far as ocular changes are concerned, in hypothyroidism, there is periorbital edema, which is seen. In hyperthyroidism, there is characteristic exophthalmos in Graves' disease in particular. We're going to see it subsequently. In GIT, as thyroid hormones, they were responsible for increasing the GIT motility. So in hypothyroidism, the motility is decreased. So there is constipation and there is decrease in appetite. In hyperthyroidism, there is diarrhea, but there is increase in appetite because more and more calories are being burned and the appetite is actually increasing. However, these individuals, hyperthyroid individuals, are actually quite thin and lean because their protein is being broken down, their energy, the calories are being burned. So in spite of the increase in appetite, they are actually losing weight. The musculoskeletal system in hypothyroid individuals, there is characteristic hypothyroid myopathy, which is characterized by proximal muscle weakness. These patients, they are prone to develop carpal tunnel syndrome and actual edema of the muscles. Hyperthyroid individuals, there is thyrotoxic myopathy, which is characterized by proximal muscle weakness as well. These patients, they are also prone to develop osteoporosis because there is increased resorption of bone created by thyroid hormones, and because of that, they are at the increased risk of fractures. Reproductive system in hypothyroidism, there could be heavy menstrual bleeding. This is a paradoxical effect. That is how you can remember it actually, that in hypothyroidism, what is happening there is chances of heavy menstrual bleeding. There could be decreased libido and infertility. In hyperthyroidism, oligomenorrhea or amenorrhea can be seen. The effects of hypothyroidism on CNS could be hyperactivity. These individuals, they are usually hypoactive, they feel lethargic, fatigued, there is weakness, they may present with depressed mood, and their reflexes would be slowed down. So one of the clinical tests for hypothyroidism is that the deep tendon reflexes can be assessed, and actually in hypothyroidism, these reflexes would be delayed or slowed. In hyperthyroidism, these individuals, they may appear hyperactive, restless, and they are prone to develop anxiety-like features, insomnia, fine tremors, and their reflexes could be brisk if assessed. The CVS effects are actually quite marked. In hypothyroidism, they may present with bradycardia and dyspnea on exertion because of the decreased cardiac output. In hyperthyroidism, tachycardia, palpitations, dyspnea, arrhythmias, or hypertension could be found because of the increased aspects of CVS functions. So now hyperthyroidism can present as a primary disease or a secondary disease. Primary disease means that the problem lies at the level of the thyroid gland itself, and it is producing increased amounts of triiodothyronine and thyroxine. Secondary means that the problem is not actually at the level of the thyroid gland. It is at the level of the anterior pituitary. It is producing more and more amounts of thyroid stimulating hormone, which in turn stimulates the thyroid gland and increases the production of T3 and T4. So causes of hyperthyroidism could be many. We are going to discuss the Graves' disease and thyroid adenoma. So Graves' disease is another frequently assessed concept of your exams. So we are going to see the details of it. What exactly is Graves' disease? It is the most common form of hyperthyroidism. It is an autoimmune disease in which the antibodies called thyroid stimulating immunoglobulins, these antibodies are actually formed in Graves' disease, and these antibodies, they act against the TSH receptor in the thyroid gland. So their name indicates that these are thyroid stimulating immunoglobulins, and they form against the TSH receptor in the thyroid gland. They bind with the same membrane receptor that bind TSH. So once these antibodies, they bind with the TSH receptors, which was supposed to bind with the TSH hormone, but now these antibodies are binding with it instead. There is going to be continual activation of the cyclic AMP system of the cells, and this is going to bring about those effects we had discussed previously, and it is going to result in the development of hyperthyroidism because of the increased production of T3 and T4. Apart from that, the TSI antibodies, these have a prolonged stimulating effect. So these TSIs, once they bind with the TSH receptors, they bind for a longer time, and they keep on increasing the biosynthesis of T3 and T4 hormones. So they have a prolonged stimulating effect. As a result of that, there is going to be increased T3 and T4, and elevated T3 and T4 levels are then going to suppress the secretion of TSH hormone. So this picture is important. Once we do the thyroid profile or thyroid function test, which we assess the plasma levels, serum levels of these hormones, we can actually find out that in Graves' disease, T3 and T4 levels will be elevated. Because of the negative feedback effect, the TSH levels will be actually decreased. So this concept is important for your questions. Sometimes in the MCQs, these findings can come in handy. So the diagnostic test of hyperthyroidism, thyroid profile, if we do in the Graves' disease, we are going to find increased total T3 and T4 level. Total here means that the hormone which is free and the hormone which is bound. So that is total. So it's actually the free hormones which are bringing about the effects. So we are going to see both of those in the thyroid profile. So free T3 and T4 levels will obviously be high. However, the TSH levels will be decreased. And you can actually find the TSI and you can see that their level is increased. So thyroid stimulating immunoglobulins will be increased in the Graves' disease in particular, not in all types of hyperthyroidism. So what exactly is the treatment? There can be a surgical removal of the gland, which will be increased in size because of the continuous stimulatory effect of these TSIs. Radioactive iodine, mechanism of which we have already discussed. The effect of iodine, it can actually ablate the gland. We can go for radioactive iodine ablation. Beta blockers can be given to actually reduce these cardiovascular symptoms, which are being mediated by the catecholamine effects. So the patients of Graves' disease, they present with characteristic ocular findings, which is known as exophthalmos. Exophthalmos means protrusion of the eyeball. So this protrusion of the eyeball, it can occur in Graves' disease. It is seen in the people with hyperthyroidism, particularly in Graves' disease. There is protrusion of eyeball. Why this happens is because there is retroorbital edema and degeneration of the extraocular muscles. This retroorbital edema actually occurs because there are certain immunoglobulins apart from the TSIs which are formed in Graves' disease that react with the eye muscles and these retroorbital tissues, cause inflammation of that. So this is how this retroorbital edema and degenerative changes in the extraocular muscles can occur, and this is going to cause the exophthalmos. Another thing was the adenoma of the thyroid gland. It can present with thyrotoxicosis as well. Depends upon the presence of hot and cold nodules in it. Hot nodules are the areas of the thyroid gland which are secreting more and more amounts of T3 and T4, whereas the cold nodules, these are not secreting any hormones. So it depends which type of nodules are present in this particular cancer and what is the level of T3 and T4 hormones. So let's discuss the hypothyroidism. Hypothyroidism, the two main causes are dietary iodine deficiency, which actually occurs in the endemic, results in endemic goiter. And then there is a thyroiditis, another autoimmune condition, which is going to result in hypothyroidism instead of hyperthyroidism, and it is known as Hashimoto's thyroiditis. So let's discuss the dietary iodine deficiency. As we have seen that the iodine is the important substrate for the thyroid hormone synthesis. When it is deficient, the amount of thyroid hormone synthesized will be significantly reduced, resulting in the endemic goiter. So why it is known as endemic? There are certain areas in the world where the soil is not rich in iodine. For example, in the mountainous areas, here the people, they are prone to develop this endemic goiter. So goiter means the enlargement of the thyroid gland, which can increase to massive size to such an extent that it can actually obstruct the airways. So lack of dietary iodine, because of that, there is reduced synthesis of thyroid hormones, because of which TSH levels are high. So in this hypothyroidism, TSH levels are going to be high because there is reduced synthesis of T3 and T4. In colloid goiter, apart from iodine deficiency, these abnormalities could also be found. There could be a deficient iodide trapping mechanism. These are some other mechanisms which can result in goiter. There could be a deficient iodide trapping mechanism. All of these mechanisms are going to decrease the synthesis of T3 and T4. A deficient peroxidase system can do that too. Deficient coupling of iodinated tyrosines in the thyroglobulin. There could be deficient organification. Deficiency of the deiodinase enzyme, so that the levels of T3 in the tissues could be decreased because of that. In hypothyroid individuals, especially who have almost lack of thyroid hormone, a condition known as myxedema develops. It is characterized by this puffy face. There is marked edema. Why does it develop? In the absence of thyroid hormones, there is deposition of interstitial hyaluronic acid and chondroitin sulfate. The GADs, they are actually deposits in the absence of thyroid hormones. So once these are deposited, they increase the tissue gel. Because of this excessive tissue gel, there is increase in interstitial osmotic pressure. Because of this increase in interstitial osmotic pressure, more and more fluid will be retained in the interstitium, which is going to result in edema. Because this interstitium is now rich in hyaluronic acid and chondroitin sulfate, it is quite rich. So the fluid which is present in the interstitium now is not going to be able to move freely. So the non-pitting type of edema will occur in this picture. Hashimoto's disease or Hashimoto's thyroiditis can actually cause hypothyroidism. There is autoimmunity against the thyroid gland, but the antibodies which are produced, they are produced against the thyroid peroxidase and thyroglobulin. So in this picture, stimulating immunoglobulins are not formed, but actually the antibodies, these are formed against thyroid peroxidase and thyroglobulin. And these antibodies, they inhibit the functions of these proteins. So as a result, there is inflammation and destruction of the thyroid gland, and the levels of T3 and T4 will be decreased. So diagnostic tests of hypothyroidism in thyroid profile, total T3 and T4 levels will be decreased. Free T3 and T4 levels will also be decreased. However, the TSH levels will be high because of the loss of negative feedback effect. Treatment for hypothyroidism involves the ingestion of thyroxine, which can be given in the form of oral tablets. Cretinism is hypothyroidism, which is seen in neonates and infants. What happens that if during fetal life, thyroid hormone synthesis is inadequate, so these babies, they develop cretinism. It is congenital hypothyroidism. There is failure of body growth, and it is characterized by mental disability because the development of CNS, it required the adequate production of thyroid hormones. So if these are not available during the fetal life, mental disability can occur. So there is failure of growth and mental disability. It can result in dwarfism. This type of dwarfism is characterized by disproportionate growth because the soft tissues, they grow, but the skeleton, it does not develop adequately. So these types of neonate who have cretinism, they are, if it is treated with adequate iodine or thyroxine, it usually causes normal return of physical growth. It depends at which stage it is treated. If it is treated early on in the life, it can actually causes normal return of physical growth. However, the intellectual disability, it is going to persist. So we had discussed the antithyroid actions of excessive concentrations of iodide. There are certain other antithyroid substances which can actually affect the thyroid function. Some of these substances which you must know, these are propylthiouracil, methimazole, carbimazole, and lithium. Some of these substances, they are actually utilized as drugs to treat the hyperthyroidism. What is the mechanism of action of these particular group of substances? They block peroxidase enzyme and they block coupling of the MIT and DIT. So as a consequence, what happens is that there is decreased biosynthesis of T3 and T4. When the T3 and T4 are decreased, there is production of TSH, the release of TSH is stimulated, and this TSH can actually cause goiter. So these drugs, they can cause goiter. Then there are certain other compounds, other ions, thiocyanate, perchlorate, and nitrite ions. Their mechanism of action is that they compete with iodide for sodium iodide symporter. So if it is competing for that, it is going to cause the lesser movement of iodide ions into the follicular cell, which is going to decrease the biosynthesis of T3 and T4. Decrease T3 and T4, once again, release of TSH is going to be stimulated because of that, and goiter can occur. There are certain goitrogenic substances in the environment which are found in cruciferous vegetables, for example, cabbages and broccoli, soy products, peanuts, and certain industrial chemicals. They also act like propylthiouracil, and they can cause goiter as well. So these are some other antithyroid substances which you can remember. These are the study resources which were used to prepare these lecture slides. You can use them to study on your own as well as other material. That is all for today's lecture. Thank you. If you have any question, you can drop it in on any of our social media handles.